Let's talk a little about sickle cell anemia. Sickle cell anemia is a autosomal recessive disease, and it's sometimes referred to as homozygous sickle cell disease because the affected individuals are homozygous for the defective genes for the beta chain of hemoglobin. They have a genotype of SS, and sickle cell anemia is the most common of all the sickle cell diseases. It affects those of African ethnicity, it affects South Americans, those from the Mediterranean, and it also affects those of Indian descent. And so it's fairly ubiquitous and characterized by the occurrence of very severe episodes of bone pain. And this bone pain is intermittent and occurs as a series of crises. And the bone pain, often in the back of the hands and the feet, is termed dactylitis. And these periods of dactylitis, these periods of very painful bone pain are brought on by physical exertion, by dehydration. And this bone pain is usually the first occurrence of the condition. Screening of the general population, which is now carried out in the UK and the US, provides us with a lot of important information about the genotype of individuals at risk. In this particular chart, we see that the parents have what's called sickle cell trait. They are heterozygous for the beta gene of hemoglobin. They have one normal allele and they have one sickled gene, one sickled allele here. So they're heterozygous. And you can see that if both parents have the sickle cell trait, there is a 1 in 4 chance that the child will be born with sickle cell anemia with both the sickled alleles. There's also a 1 in 4 chance that the child will be born without sickle cell anemia with 2 good alleles. There's actually a 1 in 2 chance, or a 50% chance, that the child will be born with the sickle cell trait. In this particular slide, we can see that the normal red blood cells carry oxygen around the body in the circulation. And these regular red blood cells, these normal red blood cells that we see here, can carry oxygen to the respiring tissues and provide the tissues and the muscles with oxygen. Now, with sickle cell anemia, the abnormal hemoglobin causes the red blood cells to sickle and adopt a crescent shape, and consequently these crescent-shaped erythrocytes block blood flow to the kidneys and the spleen, and this eventually leads to massive pain and multi-organ dysfunction because the tissues, and particularly the spleen, becomes anoxic because these organs are not getting the required amount of oxygen. Now, infants suffering from sickle cell anemia often exhibit a certain failure to thrive. They, of course, suffer from chronic anemia. They oftentimes suffer from hyperbilirubinemia because of the increased heme turnover and fatigue. They're often fatigued. They suffer from palpitations. And oftentimes they suffer from gallstones, and the spleen becomes massively enlarged. One of the most common signs in infants with sickle cell is jaundice. And you can detect jaundice from the slight yellowing in the whites of the eyes. In infants at a very young age, probably the most serious condition is septicemia. Septicemia, brought on by infection. These individuals, these children are very prone to streptococcal, salmonella and Haemophilus influenza infections. So much so that this is actually the major cause of death in childhood. It's very important, given the seriousness of the condition, that we make a very prompt diagnosis, and probably the most straightforward way to make a prompt diagnosis is by taking a blood smear. And by taking a blood smear and looking down the light microscope, we can very easily detect these sickled or crescent shapes of the red blood cells here. You can see that. You can see that here. Now, antenatal screening during pregnancy is obligatory. Antenatal screening is done routinely in the United States. It's done routinely in the UK, and we can screen for a wide variety of genetic diseases, including sickle cell anemia. One of the ways we can do this is by performing what's called chorionic villus sampling, and this uses a sample of cells from the placenta to provide us with definitive evidence of the presence of the condition. And sickle cell anemia is now included routinely in these screening panels of all newborn infants in the United States. And this is really important, because if we know the infant is going to be born with sickle cell anemia, we can start immediately daily prophylactic antibiotics. Most notably, we're going to be looking at penicillin daily, oftentimes combined with folic acid. Now, oftentimes the infant, even born with sickle cell disease, will appear healthy. And the reason for this is the large amounts of hemoglobin, or fetal hemoglobin, hemoglobin F, that's present. Although as the levels of fetal hemoglobin decline and the levels of sickled hemoglobin increase, then the symptoms tend to reveal themselves. So making an early diagnosis is absolutely crucial to the prognosis. And we can diagnose sickle cell in newborn infants, in young children who are showing these painful episodes, painful swelling of the hands and the feet. And oftentimes, if the infant is jaundiced and perhaps severely anemic, there's a variety of tests that we can do to confirm our diagnosis. One of the tests we can do is isoelectric focusing. We can even perform HPLC analysis, and we can do cellulose acetate electrophoresis. And we see that happening here. With cellulose acetate electrophoresis, the blood proteins that tend to be negatively charged are going to migrate towards the anode. So the negatively charged proteins are going to move towards the positively charged electrode. The thing with hemoglobin, though, remember, there is a mutation that causes the sickling of the red blood cells. And this mutation results in a switch in amino acids in the primary structure of the protein. So much so that a glutamate is replaced by a valine. And the valine is actually non-polar. The valine is non-polar. It's less negatively charged. And because of that, sickled hemoglobin, hemoglobin S, is going to move less towards the anode. the anode than hemoglobin A, the regular hemoglobin, because it has less of a negative charge. So this is one of the nice ways that we can make or confirm a diagnosis of sickle cell anemia.